Over the next 25 minutes, I'm going to tell you about the microbiome and how it impacts health and disease. The human body is composed of 30 trillion human cells. This differs from 38 trillion microbial cells, but the key difference is in the gene content. So the human genome is made up of 20,000 genes, whereas microbial genes up to about 8 million contributed by 10,000 different species. Bacteria in the gut, as well as in various mucosal sites of the body, play an important role in health. In the gut, they break down polysaccharides. They help in the biosynthesis of vitamins. They prevent pathogens from colonizing the gut, referred to as colonization resistance, and play an important role in maturation of the immune system. When the microbial community structure, or the term we use often called dysbiosis, is altered, one is at risk for various diseases. I'm going to highlight how the microbiome is altered in diseases like inflammatory bowel disease. And broadly, we define this as microbes that are enriched or depleted. Now, when the microbiome is broadly referred to as not only the bacterial microbiome, it also includes the fungal content, which is referred to as the mycobiome, and similarly, the viruses in these various body sites referred to as the virome. But for today's talk, I'm going to focus on the bacterial microbiome, and the principles that I'm going to allude to in the bacterial microbiome are broadly applicable to the microbiome and the virome. These are my conflicts. So to get started, how does one study the microbiome? And I'm going to tell you how the gut microbiome develops over time, how they respond to perturbations such as diet, and how it changes during disease. To measure the microbiome, we use 3 different metrics. So we take stool or tissue biopsies, isolate DNA, and by amplifying the 16S ribosomal RNA, we can tell who's there, which bacteria are there. And by sequencing the genomes, referred to as metagenomics, we can tell who's there and what they are doing. Bacteria produce products, and so by, again, starting with stool or plasma, we can measure the metabolites that are made by bacteria, and this measurement is done by metabolomics. When one defines the bacterial community structure, one starts with phyla, and today I'm going to tell you about data generated by 16S or metagenomes. And so here I'm going to refer to the family of bacteria, the genus of bacteria, and the species. And then a finer resolution of the species could be broken down to many strains, and that's only possible by metagenomics. Now, there are important similarities and differences between studying microbes and studying genes. The similarities are in both settings, we want to find out high-dimensional features that are associated with human phenotype. So what's the microbiome in an obese individual versus a lean individual, in a smoker versus a non-smoker? We then consider the variation in genes, but what's unique in bacteria, we can also measure abundances. And then the community structure is then further classified by diversity, metabolites, and taxonomy. The key differences are the human genome is relatively stable, whereas the microbial genes can change with time and evolve. Transmission of the human genes are only done vertically, but whereas the microbial genes can be shaped by many environmental factors. And then when it comes to causality, while both human genes and microbial genes interact with the environment to shape a phenotype, only the microbial genes can be shaped by the environment as well as the phenotypes, so in this case, the disease. So in the next few slides, I'm going to tell you how age impacts the microbiome. And so when we are born with an abundance of the Bacteroideaceae families, as you can see from this time-dependent plot over the first 36 months of life. But then we start with very low levels of Lactospiraceae and Ruminococci that increase with time as we age and the immune system develops. The bacteria have an important role in the development of the immune system. And so one can begin to appreciate that if one were to be prescribed antibiotics early in life, that this community setting could change significantly. Now, the microbiome in various body sites differs. So you can see that the microbial content in the gastrointestinal lumen is quite different from the oral lumen. Obviously, oral bacteria tolerate oxygen, whereas gut bacteria are mostly anaerobic. And then similarly, there are key differences in the skin and in the nasal mucosa. Now, if you go at the other extreme of life, so in this study, we examined the microbiome and the metabolite content of centenarians and compared them to individuals at much younger age groups. And you can begin to appreciate that the bile acid profile, so there are primary bile acids and secondary bile acids. Secondary bile acids can be modified by bacteria, and you can begin to appreciate that these secondary bile acids increase in abundance in the centenarians. So in this study, we asked, what do these various secondary bile acids belonging to the lithocholic acid family do? And here are two examples where these metabolites made in the centenarians clearly educate T cells to make T cells more regulatory in nature. So they are mostly dampen inflammation. And they also have features of preventing pathogens such as C. difficile from colonizing the gut. So a beneficial effect of the microbiome with age. What about diet? So obviously there are many dietary regimens, but to simplify the observation, we then asked, what does a gluten-free diet do to the microbiome community structure? And here's an abundance of the change in the abundance of the Velanelaceae family. So what I'm showing you is on the x-axis, the abundance in somebody on a habitual diet compared to a gluten-free diet. There's clear depletion of this family of bacteria. And so one could extend these observations by looking at the effects of the microbiome that has been published on a ketogenic diet, a Mediterranean diet, and various diets, and quantify the changes in the bacteria over time. What about health and disease? I'm going to give you two examples. And the first was a study where 1,000 children born in Finland, Estonia, and Russia were enrolled in a study. The risk of type 1 diabetes in Finland, as you can see, is approximately 10 times that of Russia. And the microbiome from these children was collected every month for 3 years and asked the risk of autoimmune disease, particularly type 1 diabetes. And as you can see here in this plot, as you look at the microbiome developing from 0 to 36 to 24 months, if you happen to be born in Finland, you had this abundant orange band that was made of the Bacteroidetes, which was very low in children born in Russia, but they had a thicker blue band. These are the actinobacteria that remained steady in the kids born in Finland over time. So with this type of detailed microbiome map over the first 24 months of life, we then asked what's different in the functional content of the microbiome in kids born in Finland versus Russia. So if you remember from my previous slide, the risk of type 1 diabetes is significantly higher in kids born in Finland compared to the kids born in Russia. And what we uncovered was the biosynthesis of lipopolysaccharide and their genes was significantly different in kids born in the two different countries. Now, what was striking was the lipopolysaccharide made in kids born in Russia was one of these 6 acyl chain LPSs, whereas the bacteria found in the Finnish kids made the 4 and 5 chain LPS. Why does this matter? So the 6 acyl chain LPS that is dominant in the Russian kids bind to a receptor called the toll receptor 4, and they educate the immune system. And this educated immune system can decrease the risk of autoimmune disease. So the risk of type 1 diabetes is significantly lower in kids born in Russia. Whereas the kids born in Finland, this education did not happen because the different structures of the LPS and this poorly educated immune system increased the risk of type 1 diabetes. So an example of how the early life microbiome plays an important role in educating the immune system and altering the risk of autoimmune disease. So are we losing our friends, and the increased uptick in allergic disease, is it mediated by the gut microbiome? Now, there are many diseases that have been identified and associated with disease. Another way of looking at this is examining this plot. So what I'm showing you are publications related to particular diseases on the x-axis in the different years and the cumulative PubMed entries. And you can see a marked explosion across many physiological states and disease states and microbiome association studies. So let's apply this to one particular disease, so inflammatory bowel disease, which is composed of Crohn's disease and ulcerative colitis. I don't have to tell this audience about the differences and the commonalities between the two diseases. But in a study where 28 North American pediatric hospitals enrolled 450 children with newly diagnosed ulcerative colitis, and their biospecimens were collected before treatment was started, and they were prescribed the standard of care treatment. So most of them were started with sulfur-based drugs, particularly mesalamine, and the kids with more active disease either received corticosteroids and, when needed, anti-TNF drugs. So you can see in this plot, what I'm showing you, features of changes in disease severity over time compared to what happens to disease remission activity. And the take-home message is that if you happen to be born with mild disease, at the end of 1 year, 37% of children achieve remission. Whereas if you were born with moderate to severe disease, the upper panel and the lower panel, you can see they achieve only a small subset, 14% achieve remission, and 49% had refractory disease despite the standard of care treatment. If you look at the various abundances of bacteria, so you can look at bacteria that are increased and decreased in patients with ulcerative colitis. Clearly, there are the bacteria that increase with severe disease. I've listed here and belong to the Enterobacteriaceae family. There are certain oral bacteria such as Haemophilus and Veillonella that could be found in patients with severe disease, whereas there were many so-called healthy bacteria that were markedly depleted. What this study then also allowed us to do was, by comparing the microbiomes at the time of diagnosis and 4 weeks later, be able to predict who would have a colectomy. And I'm showing you here the fold change in bacteria that is associated with risk of colectomy in this pediatric cohort. So predictive value of the natural history of disease. So in the second disease example, I've told you that with the current modalities of treatment, about 30 to 40% of kids achieve remission, and that depends on the severity of disease, so suggesting that there might be an important role for the microbiome, and that the baseline microbial signature could be important in predicting refractory disease versus the need for a colectomy, suggesting that one needs to pay attention to the microbiome in optimizing treatment outcomes. And then the fourth outcome is, how does patients respond to treatment? And as you can see in this picture, the microbes affect various dietary compounds, various pharmaceutical drugs we might consume, and then, as I already mentioned, bile acids, but it could metabolize many of the metabolites that are made by the human body. And so I'm going to tell you how drugs influence the microbiome and how microbes influence the drugs. And in this example, I'm going to highlight the sulfasalazine drugs. So we are aware that sulfasalazine, when consumed, is in an inactive conformation, and in the gut, in the colon, this is broken down into sulfapyridine and the 5-ASA active moiety. We also know that in a subset of patients, about 50% of patients fail to respond or lose response to these 5-ASA drugs. So in this study, we asked the question, are there microbial features that would tell us about who's going to respond to 5-ASA drugs and who's going to lose response? And so by enrolling 2 cohorts of patients who used the 5-ASA drugs compared to those who did not use the drugs, we sequenced the microbial genome, we measured their diet, we checked their metabolites that are produced, and then tried to associate microbial gene content and metabolites with 5-ASA use. And what we uncovered was this class of enzymes called the acyltransferases that were involved in inactivating the 5-ASA molecule. We could confirm in vitro in a test tube that this is true, so the predictions could also be confirmed. And then by studying an independent data set, we could show that the same as 4 acyltransferase enzymes could be associated with presence of the 4 acyltransferase enzymes could be associated with increased risk of treatment failure for 5-ASA drugs. So the benefit of these types of studies is could we come up with a predictive tool set of the microbiomes that are associated with severe disease, failure to respond to drugs, and developing drug resistance. What about extending this to cancer? So similarly, could we identify microbes and microbial metabolites associated with cancer? And in this study, the authors examined 7 different cancers, looked at their stool microbiome. And what I'm showing you with the panel on the right side is the relative abundances of bacteria in pancreatic cancer, glioblastoma, all the way down to colon tumors. And you can see that there are clear differences in the relative abundances of bacteria, while some bacteria are shared between various types of cancers. So with this foundational study, we then asked, is there patterns in the microbial content as polyps evolve? So from normal mucosa to early adenoma to late adenomas and cancer. So the gene pathways that are associated with adenomatous change have been well studied. And we wanted to see whether we could then map the same microbiome features with gene activation, such as APC activation, the presence or absence of a RAS mutation, and P53 mutations. Analyzing 2 large data sets, we could identify that there were specific microbial signatures or microbial abundances that are associated with tubular adenomas compared to serrated adenomas. And the functions of these microbes also differed. So the tubular adenomas basically had microbes that had altered metabolizing capacity of methane, whereas the serrated adenomas had increased ability to modify bile acids and sulfate metabolite potential. So clearly, bacteria could match the gene content changes that are associated with polyp progression. What about cancer? Again, looking at colon cancer, a number of bacteria that have been identified. I'm going to focus today to highlight the Fusobacterium nucleatum family. So in this particular study that was published earlier this year, they examined the presence or absence of Fusobacterium nucleatum strains that are normally residing in the oral cavity and those that are associated with colorectal cancer. And they were able to identify the microbiomes that are associated with colorectal cancer. So getting back to that classification of these were metagenomic data, so we could tell something about the strain level annotation. And what was striking is that the colorectal associated microbiomes of the Fusobacterium nucleatum family was very different from the Fusobacteria that live in the oral cavity. And if you then isolate these C2 enriched Fusobacteria and then give them to mice with increased susceptibility to colorectal cancer, they ended up developing large intestinal adenomas. And these authors then, in an independent data set, also confirmed the presence of the different strain of Fusobacteria, so in tumors that are associated with colorectal cancer. So an example of why finer classification of the microbiome is important as we get to causality. So I've told you examples of how the microbiome is altered in a disease like type 1 diabetes. I've given you an example of how it might impact a disease like IBD, its impact on disease course, treatment response, resistance to drugs. And then in the last example, I've told you how the microbiome could be matched to the progression or the development of adenomatous change in the gut and the unique features of the microbiome associated with colorectal cancer. The good news of all of these types of scientific observations is that now we are at a position to try to identify predictive tools as well as tools that microbial abundance, gene content measurements that could advance diagnostic aids that could be used for these diseases. Very briefly, what about the liver? Yes, there are many associations of microbiome with liver disease, changes both in the bacterial microbiome, the virome, and the microbiome have been described in patients with liver disease, and the same principles apply. Microbial genes and metabolites affect the progression or the resolution of disease in the liver. And here's an example of why this might happen. So the gut has two large channels of communication between the liver and the gut. So the bile duct is an opportunity for bacteria and metabolites to go up and down. And similarly, the metabolites that get across the gut barrier get into the portal circulation and first meet the liver. So in this study, authors asked, are there features in the bacterial microbiome and the microbiome that's associated with alcoholic liver disease? And I'm summarizing the key findings here. The abundance of a certain toxin producing E. faecalis correlated with disease activity in patients with alcohol-induced liver disease. Similarly, there was these candida organisms that secreted a specific product that increased the inflammatory response by priming these cells called Th17 cells. And then the depletion of bacteria that make short-chain fatty acids called butyrate also reduced the protective barrier function. So you can see that butyrate plays an important role in holding epithelial cells together as a tight junction, and that gets altered and makes it more leaky when a product such as butyrate is depleted, so thereby allowing communication or free flow of information exchange between gut microbes, gut fungi, and the immune system and the liver, which is sort of a collection agency when it comes to flow of information. To touch upon treatment, obviously there's been rapid progress over the last few years of the impact of the microbiome. The first landmark, very briefly to get back to a question that I'm often asked, are probiotics helpful? The challenge is, as you can see, no randomized trials have shown the effects of these probiotics on many of the features listed here. But more importantly, the bacteria that I've listed today that are increased or decreased in abundance are rarely in a probiotic capsule. So with the new information that we've collected, I think one needs to go back and redesign better microbial therapeutics. So the success story was with C. difficile colitis. So patients who develop C. difficile colitis more or less markedly deplete the gut microbiome, and estimated efficacy of antibiotics for first recurrence is about only 60%. Bacteria, C. difficile toxin, this is a study that we recently identified that the effects are primarily on the sensory neurons. They secrete stimulation of neuropeptides that makes cytokines, and clearly showing how a toxin made by C. difficile alters the immune status of the gut. So in a landmark study, patients were treated with vancomycin. They were then treated with vancomycin for 4 days of that gut lavage and then a donor fecal microbiome transplant, and asked, what about the effects of the antibiotic treatment compared to the fecal transfer? And as you can begin to appreciate, the donor transfer was associated with a significant response and a marked lowering of the recurrence rate. And you can, if you look at the gene content of the microbes that were transferred in the FMT, you can see that many of the donor microbes are found in the recipient who received that FMT. This study was followed up 2 years ago, where the microbes were now presented rather than in a fecal microbiome transplant in a capsule. And the question was whether this series 109 could be used as a more purified prep to try to correct C. difficile colitis. And the findings were similar. So clearly, treatment reduced the recurrence rate. The recurrence rate was maintained across age ranges. So if you compare the dark blue to the light blue bars, the recurrence according to a previous antibiotic, again, was significantly influenced by SER 109. And then you can begin to appreciate how the bacterial composition changes in individuals who received the capsule with the microbes in it. So there's a real opportunity to try to impact many diseases using this microbial therapeutics approach. But then one could also think about delivering the metabolites, the products that the bacteria make, as another therapeutic strategy. So to summarize, in a disease such as fecal C. difficile colitis, you have a disease state that's clearly identified. And this is also true in conditions like graft versus host disease, or in the clearance of MDR bacteria, the decolonization of MDR bacteria. And successful studies have shown that conventional fecal microbiome transfers can correct the phenotypes from disease to health. Now, the more challenging outcome has been in diseases like inflammatory bowel disease, which fluctuate over time, where you can get transient correction by fecal microbiome transfer studies, but then if you don't control the underlying inflammation, patients then relapse back. So going forward, I think what's needed is to try to combine inflammation control with microbiome transfer control or microbiome-based therapies to correct diseases such as inflammatory bowel disease. And this is also true for metabolic syndrome, irritable bowel syndrome. So what these studies that I've highlighted today hopefully point to is that we now have good tools to measure the microbiome. We can begin to appreciate who's there and what changes with disease activity, how the changed microbiome impacts resolution of inflammation in a disease like inflammatory bowel disease or progression of polyps to colorectal cancer, and then how the microbiome might impact various standard of care therapies. And I've given you an example in detail about how 5-ASA drugs could be altered by the microbiome. And then more in the last example, I've given you the principles behind fecal microbiome transplant treatment for diseases such as C. difficile and what might be a path going forward. I want to end by highlighting an example of how the microbiome could be used as a predictor of disease. So this is a cohort of individuals who underwent allogeneic hematopoietic stem cells transplantation. These studies were done in 4 different institutions: Memorial Sloan Kettering, Duke, in Norway, as well as in Japan. And the first bar plot shows you months over proportion surviving as a result of the HCT transplants. You can see that if you had high bacterial diversity to start with, you did a lot better. Survival was significantly improved. And this was true in all 4 sites. Higher the diversity, the better the allogeneic HCT treatment outcome. They could also show that transplant mortality, again, was associated with bacterial diversity. So this was a term that I introduced early on. You can measure the gene content, but you can also measure how diverse the microbiome is. And here you can begin to appreciate that the higher diversity individuals monitoring months after day 21 clearly has an impact. And then graft composition is also influenced by microbial diversity. So you can look at T cell biology and cumulative index over time. And you can, by comparing the red versus the black line, you can clearly see differences of how the microbiome could be used as a predictive tool in allogeneic HCT, and this types of studies could be extended. And finally, I'd like to highlight how the microbiome might impact micronutrient deficiencies. So there are detailed epidemiological studies that vitamin D deficiency is prevalent in the US, and it's linked to insufficient exposure or insufficient consumption. And dietary vitamin D consumption or loss is mediated by this globulin that increases or decreases the bioavailability of vitamin D, or the conversion of these metabolites to 25-hydroxyvitamin D or 1,25-hydroxyvitamin D. 1,25-hydroxyvitamin D then binds to the vitamin D receptors. And what these authors were able to show is that micronutrient receptor interaction promoted the abundance of a specific bacterial strain in Bacteroides fragilis. And the presence or absence of Bacteria fragilis had a marked impact on how these tumors responded to checkpoint in immunotherapy. So obviously, the initial studies were done in mice, and the observations were then confirmed in a human cohort. So what I've told you in the last 25 to 30 minutes is how one can begin to appreciate the impact of the microbiome, and how one might measure the microbiome and the health benefits of the microbiome, and a peek into how the microbiome might impact disease course. Thank you very much.